Now we're shifting gears a little bit, and we're talking about prediction of response to actually an established drug. Just to start out, checkpoint inhibitors are exploited by cancers in order to evade the immune system. And checkpoint inhibitors, they are an antibody-based therapy that actually impairs this evasive mechanism in order to reinvigorate the immune system so that the immune system can kill the cancer. This has really improved the treatment of several solid cancers, and an increasing number of approvals for new cancer types are coming every year. So it is becoming quite widely used and also as first-line therapy. Now, since this has really improved the therapy of several cancer patients, it is also reflected in the market. And Christian already alluded to that the market for checkpoint inhibitors are already now fairly large, but it has actually been projected to reach all the way up to 150 billion USD by 2030 worldwide. So this is a big market. However, despite this being a clear improvement in patient care, then as Thomas also talked about in the Pioneer session, a large fraction of patients actually still do not benefit from the checkpoint inhibitor therapy. And that, of course, is an issue. So there's really a demand for increasing or further development to increase the efficacy of the immunotherapies, but there's also a demand to identify those patients that do not respond to therapy. And that's also namely because as good as this therapy actually is, it also can cause quite severe side effects in certain patients. To the physician's frustration, however, there are no established biomarkers which are able to predict which patients actually do respond to the checkpoint inhibitor therapy. So that really underlines the issue here. So now I just want you to remember back to the previous sessions where Thomas nicely described how Pioneer is able to quantify the quality of the new antigens and how that correlated to the progression-free survival of the patients in our in-house combination therapy trial. Furthermore, we also have the observed-derived biomarker, which we saw in the last session actually is predictive of the overall survival of melanoma patients receiving checkpoint inhibitor therapy. So we already have established that biomarkers derived from our 2 models for personalized cancer vaccine development actually can be repurposed to biomarkers predictive of patient response. So we thought that we are in a pretty good situation here and have sort of a responsibility to develop a new model to help the physicians identify those patients that do not respond to the checkpoint inhibitor therapy. So basically, the flow would be that you have a cohort of patients, some would be responding, some would not be responding, and you have to identify those that respond. That would be based on a tumor biopsy, which the doctor would collect from the patients, and then a genomic profile of the tumor biopsies, which would then be analyzed by an AI model to discriminate between the responders and non-responders. So basically, this has to be done through the lens, in our view, of genomic biomarkers. And this has several benefits to the patients. If you can identify a patient that you know will not respond, then the patient can be redirected to an alternative therapy that could have a potentially a higher chance of providing the patient with a benefit. Furthermore, the patient would also avoid the risk of severe side effects. And of course, there's also an issue that is talked about now, the impact on the healthcare budgets. So you could alleviate that as well. by avoiding to treat patients that do not benefit from the treatment. So the genomic biomarkers involve driver mutations, the environment of the tumor, the immune evasiveness of the tumor, also different sources of antigen burdens, and of course biomarkers derived from our pioneer and observed model. So put in the phrasing of the AI-Immunology platform, we basically take certain elements from the AI-Immunology platform in order to design our predictive model called AI-Deep. For developing the model, we then collected a lot of genomic data from 937 cancer patients who received checkpoint inhibitor therapy. The data was comprised genomic profiles of biopsies collected prior to the treatment. And we then used this data in order to identify the most predictive set of biomarkers, which was then advanced onto the final model development to really see if we can predict patients that have a clinical do not respond to the checkpoint inhibitor therapy. Yeah, so what we then saw was that if you look at the traditional biomarkers here displayed in the figure with the red curve, then you can see that they actually fail to achieve a very, very high precision, which would be warranted in order to avoid depriving a patient for potentially curing therapy. On the contrary, you can see that the AI-Deep model is actually able to achieve the high precision that is wanted, and it does so for 28% of the patients that do not respond to therapy. So all in all, AI-Deep could actually aid the physician in taking treatment decisions. So furthermore, we wanted to investigate, so now we have a range of really predictive biomarkers, but which of them is the most predictive biomarker? So we did that by conducting what is called a feature ablation study within the field of machine learning. And that is basically a study where you estimate the importance of each of the biomarkers on the predictive performance. What we then discovered was that our in-house developed biomarkers derived from the pioneer model and the observed model actually ranked among the most informative biomarkers, whereas the more established biomarkers, the tumor mutational burden and the PD-L1 expression, actually ranked as being much less informative. So in summary, we just want to list here that AI-Deep leverages our pioneer and observed model in order to predict whether patients will respond to checkpoint inhibitor therapy, and it can do so quite accurately for a subset of the non-responding patients. And we're currently exploring opportunities for a commercial offering and also further clinical validation as a companion diagnostic. Thank you.